Hello, everyone. Welcome to today's webinar featuring Ian Wilson, CEO of ImaginAb, a company at the forefront of imaging and radiotherapeutics. I'm Echo Hindoyang, and I will be your host for today's discussion. I'm a CEO at MSQ Ventures, a New York-based advisory firm specialized in life science. We are excited to host this webinar interview with Ian and our New York office today, and even more thrilled to meet in person after being virtual for so long. We are excited to hear from Ian about the latest developments in this exciting field. Throughout his career, Ian has held a number of significant healthcare roles and brings a wealth of experience to his current position. In this interview, we will dive into ImaginAb's technology, pipeline, partnership, and future plans, as well as its perspective on the biotech industry as a whole. So let's get started. Welcome in. So nice to see you in person.
Hello, Echo.
Hello, everyone. So we want to just make sure everyone knows we are in person together next to each other. Even now we are on the two computers.
Yes.
So please tell us about your background and how you come to be the CEO of ImaginAb.
Thank you, Echo. Yes, I've worked in radiopharmaceuticals for the majority of my career. I even studied imaging at postgraduate level, and I started my career at a company, AstraZeneca, doing magnetic resonance imaging, and then had a long career at GE and GE Healthcare for nearly 20 years. So I was there, and that gave me a good foundation in learning all aspects about imaging and radiopharmaceuticals. When I left at GE, I started working for small startup companies, and that's where I gained my experience of fundraising and moving molecules through the gain value and potential return on investment for investors. I actually then joined ImaginAb just over 5 years ago. I first came across ImaginAb when actually I worked at GE. We were very intrigued about the fundamental technology that ImaginAb has, and I'll talk about that later. So it's been a great journey with ImaginAb. We are a company that is leading in a specific field of immuno-oncology imaging, and I will explain today that we're also now working in a very exciting field of radiopharmaceutical therapy. A lot of people have interest in this because of the work and acquisitions by Novartis and Bayer, bringing therapeutic drugs and a new therapeutic modality to the industry.
Thank you. I think the most impressive one is actually you joined ImaginAb in February 2018 to be the COO, and then just in 1 year you were promoted to be the CEO, and then by the most difficult time, the second quarter of 2022, you made ImaginAb to be the profitable or revenue have the revenue and also the self-funded biotech company. I think this probably is a very valuable lesson for all the biotech executives for today.
Yeah, thank you, Echo. It was by need and design why we went that approach. One, because actually I started the journey with ImaginAb, actually radiopharmaceuticals weren't in favor with investors. They were interested in investing in other parts of the industry. So we identified that and thought, how could we actually get revenue? And I'll explain that we actually I've been generating revenue since 2018, and I've had year-on-year growth of greater than 20% per year as we do that. So that's offset our capital raising needs. Yes, so as we say, for every dollar that we raise by investors, we bring a dollar to the table from our revenue, which makes our R&D very cost effective for our current investors. Yes.
Yeah, thank you. And also, you can go deeper later during your presentation. And I think also another thing very unique of the ImaginAb is ImaginAb have been partnered with a very diversified group of the like a biopharma, CRO, and also with the different regions, South Korea, Turkey, China, and the US and Europe. So could you please give your background? What's that? How did you develop this strategy? And also, to make one deal happen, transaction happen, it takes so long. But however, in the past few years, I read your news like every month, you have the news announced, the new agreement signed.
Yeah, so yeah, you're right. Deals do take a long time. They take on average at least a year. One, because of negotiation sometimes with some companies is complicated and it involves lots of stakeholders. In the start, each of our deals was very bespoke because our technology was emerging. So we had to, work with each partner to come to terms. But as we did more and more deals, we started with large pharmaceutical companies, the ones that saw an advantage of implementing our technology into their clinical development plans. And then in the last 2 years, there's been a switch towards more biotech companies using our technology. And with that, we could actually just generate template agreements. So because really each agreement, whether you're big or small, it's the same with us. We don't price people differently because of who they are and where they are. We have the same price point for all our customers. So now we do everything in a very turnkey fashion. We have the same deals. Our expansion around the globe was really because we had to undertake our clinical trials, but at the same time, we knew that the pharma companies and biotech companies wanted to do trials in global locations. So although companies are maybe headquartered in the US, they may want to do trials in Europe and Australia and vice versa. So we started off with establishing manufacture with our partners in the United States, closely followed by Europe and Australia. And really that's where the majority of our studies are. As you mentioned, we've highlighted South Korea. A lot of people are interested in South Korea, so our plans is to open in South Korea, both because of clinical need for our tracer, but also pharma companies want to work there. And I think the greatest decision we made was actually to work in China. And that's where we connected and we went on a tour of China. And thanks to you, we understood the Chinese marketplace. The fundamental deal that we eventually did was based on the understanding we had when we went to China. It's very difficult for a company based in the US to work effectively in China. Yeah. So we knew we had to partner and out-license the technology and have some people on the ground. So that's how we subsequently did deal with Dongcheng or DC Pharma, who took rights for our technology in mainland China, Hong Kong and Macau. And they actually see the same market. One, working with Chinese-based pharma companies who want to advance their immuno-oncology therapy, but also see that they can get it approved by the Chinese authorities for use as a broad medical imaging product in clinical decision making.
So could you please go deeper about also the diversified group of the biotech, biopharma, and the CRO, because you partner with a big pharma like Roche, and then you also partner with Dongcheng, like a radiotherapeutics company in China, but you also partner with the CRO, and so I think it's like you probably partner with the companies or in the value chain. Yeah. So what's that angle, and how do you manage the different dynamic of the relationship?
Yeah, so fundamentally we're an R&D company, trying to get our products to approval. So, but we have to have a sales and marketing team to actually promote our technology to those pharma companies and sign deals. And so you have to balance this time you spend on R&D and the time you spend on finding money and generating money. And so that's why we did the deals with the CROs, because we can only touch so many people. Yeah. The CROs talk to the pharma companies on a daily basis. Oh, okay. So actually, by signing agreements with the CROs for them to enable them to promote our technology and sign deals directly with them, it's like adding another layer of a sales team to us without us investing. So the CRO approach was a way for us to explore a different sales channel. So they actually sell and promote our technology and can package it in other pieces of work that they do with the pharma companies. And some pharma companies actually have multi-year deals with single CROs. So if for them, it's an advantage to offer that, have another product offering to keep that pharma company.
So it's kind of like have the incentive for the different level of their clients.
So in that case, it becomes very simple. We just become a supplier, and the CRO has to do all the contracting, and we just have a supply agreement with those CROs. So it makes it very simple for us. Regarding the diversity between large companies and small companies, fundamentally, they're all using it the same. They want to be able to understand if their drugs work, and if they don't, stop working on them, and if they do, advance them. So really, we provide a tool that's used similarly between each of those companies. The biggest difference is that the larger companies may do multiple projects with us, while smaller biotech companies probably may only do one followed by the next. So that's the biggest difference. And then the other difference is probably what they're actually testing. In immuno-oncology, there are many different types of drugs, checkpoint inhibitors, vaccines, cell therapy, and that's where we see diversity is in what drugs they're using with our agent.
So your goal is as many as the patients can access to your technology, and through the different partners. That's a goal.
Yeah, our goal is one was to get revenue, but secondly, we're starting to get our technology used. And of course, although pharma companies do the license, clinicians are using it on their behalf. So they get interested. So we are already getting our agent known by clinicians, which is very useful before we get approval. Secondly, in our deals with the commercial companies, we get access to safety data. Which means that we actually don't have to collect or do as much trials as ourselves. So we have an offset of expense there that we don't actually have to do those safety studies ourselves. Oh, we do, but we don't have to collect as much data. So both they are bringing data to us. And, that is the hope. And that is the hope that it will hopefully accelerate our approval, firstly with the FDA and then in other markets.
Actually, you already made the big milestone last year. You already have the first patient enrolled for the phase 2, the CIT nano trial, right? Last June. And so how that progress now?
Touch wood, it's progressing very well. That we've got a very good clinical operations team, and kudos to them. Like everything else, we enrolled that trial. We may have had and the recruitment is going really well, so it's on track. We hope to complete that trial in 2024. And that data will be pivotal for our submission to the FDA.
Wow, congratulations.
Thank you.
So as we know, you collaborate with MSK and MRA on this trial, right?
Yeah, so we've actually got over 15 sites undertaking the trial in the world, and we do trials ourselves in the US, Europe, and Australia. So as an example,
so it's a global trial.
It's a global trial, and so one of the indications we're looking at is melanoma. So of course Australia has a high incidence of melanoma, so it was natural to go there. And so we see that. So we although it's for the FDA, we've opened multiple centers across the world. So including ourselves, the sites that we use, and pharma companies, there's over 65 sites globally using our technology in those 3 continents and growing. So, that shows that, there's many key sites, including, as you said, Memorial Sloan Kettering, and big centers like the City of Hope, but then down to sites less well known, but great for us, like, Little Rock in Arkansas. People go, where's that? I do, as being British, I have to look it up on that. But they are a great recruiter for us and great. So we have a great geographical spread and great clinicians using our technology.
So I think that is kind of like you have been planned, the region and the different layers of the partners. So why do they choose ImaginAb? Another question is should be, how does ImaginAb's technology different from the other like imaging or radiotherapeutics current on the market?
Yeah, so luckily we were first to the market and currently with the only clinical compound that can do what we do. So that's always a good place to be. So it's a good place to be, and it's a good place to stay ahead of the competition. There are other people now developing tracers, which is good for us, showing that we want to do it. The key reason, I'll show it in the presentation. The key reason is that our technology gives them more information and actually gives them an answer maybe quicker than standard technology. So, if you look at the cost of clinical trials per month or per patient, if you can make savings, such as allowing someone to make a decision 1, 2, 3, 4 months earlier, then theoretically it will save them money because they can terminate the trial earlier on positive or negative results and move on.
Yeah.
So that is the current value proposition, and how it's used by pharma is very similar how we think it will be used in a clinical setting. It'll be used to determine whether the treatment that someone's on is correct and whether they should receive one monotherapy or combination therapy to help them evolve.
Yeah.
So that's how we use it. Our first entry point was as a replacement of biopsy. Biopsy is difficult in clinical trials. Some of the pharma companies identified that, and that's how we sought our technology, that it would allow them to negate the use of multiple biopsies of multiple tumors in metastatic patients. And that's how we started. And then we have grown the technology and discussed pharma about what kind of extra data they want, and we've built that into our development plans so they get more confident as our technology evolves.
That is the value population. So we have been talking about all the positive things. I think the success you made and how it sounds like very easy, but we understand that there's a lot of challenge behind it. So what are the challenges you have been facing in bringing ImaginAb's product to the market, especially actually is still developing, but you already made a cash positive from this product?
Yeah, it's as someone says, it is the art to look like a swan gracefully, but you don't see the legs kicking under the water, right? I think that's what people have always said that to us. ImaginAb is doing a great job, but at the end of the day, we've got a fantastic team who are very dedicated, right? And we use every dollar that we have wisely, and it's just simply that. It's good financial management and just focusing on the key things that we need to do, so not getting distracted, right? So we've always spent our money wisely, and that has been very key in the last few years because of the markets. One, because of COVID, and recently people are very cautious about fundraising and getting money in. So I think we've always had that strong ethos of being careful with every dollar that we spend, and I think that's put us in good stead for where we are today.
Actually, you kind of you just talk about the COVID. I think when you started the strategy, when we back to 2019, we went to China together. You or just started to this plan, and then it's just really this kind of developed so quickly in the past 3 years. And you just have been done all this, get this done in your house.
Yeah, so in the US, because we're an oncology company, US law allowed us to still be in the office and the laboratories. So we still had lots of people coming to the office and working.
During the pandemic,
which was good. But those that weren't essential for the laboratories stayed at home. We did see a downturn because a lot of clinical trials themselves suffered from not being able to undertake clinical trials. But post-COVID, you would expect most people were at an urgency to catch up. So, and that was beneficial for us because they wanted to speed up their clinical trials and try and gain lost time due to COVID. And that's what we saw. We saw a lot of the drug companies then say, hey, during COVID, we want to use this technology. Let's sign it. Let's use the downtime to agree deals. And we agreed deals during COVID, enabling them to use the technology post the COVID pandemic.
Well, it seems the technology in terms of what you mentioned about can really help the biopharma biotech company actually save the money for even the drug development stage. So I think that's why you are not only survived the pandemic, but you actually thriving because people see the benefit of partner with the ImaginAb.
Well, yeah, we hope that, I think, to say that we save money is correct, but really, do you really, when you pay for something extra, you're actually asking them for more money.
Yeah.
So it's very difficult to go in a question about money saving. It's really about that they see the technology will help them answer the question they need to. And maybe it saves money in the long term because they design the next trial correctly. They understand where they have to do mono or combination therapy. So we don't sell our technology saying we're going to save you money now. The point is we help you make a decision.
Go or no go.
Go or no go.
Yeah.
Or whether to change the strategy of your progress. So such as, did you pick the right dose? Did you pick the right cancer indication? Did you pick the right dosing regime? Should it be drug 1 first or drug 2 first, etc.? That's really it. And for some who are in the early stages of new technology, vaccines and cell therapy, it's really about, we've seen really good results preclinically. Actually, we'd like to see if actually that translates into the clinical situation.
Yeah.
So for them, it's really about proof of principle of their technology and being able to show pictures to investors and clinicians and other people to gain interest.
It kind of increase the success rate of the
I hope
trial.
so.
That's the most expensive part of the whole drug development, right?
Yeah, as you say, a picture's worth 1,000 words, and I think some of our images, they speak for themselves, and people can show their technology works at all level of a business, from people at the laboratory all up to the boardroom. They can show our technology to them, and it's easy to understand how it could make a benefit.
Yeah. So I wanted to just take the up level, because you have been working with the partner around the world, right? So what's your, what do you see the landscape of the radiotherapeutics development now?
Okay,
globally.
So switching to radiopharmaceuticals as therapy, I think there's been a great interest. It's one of those technologies that's took many years to get there. It was really driven by academics. It took 20 to 30 years of evolution, and certain things had to come into place, like as we talked about, is there a global infrastructure, is the isotope supply, and now it's not 100% sorted, but people can have confidence that they're similar to a cell therapy, which is a logistic decision. Radiotherapies is the same. You have to make the compound, distribute it very quickly to the patient, and get it used. So there is a complex supply chain. If people are okay with that, then it's a great technology to work in. So radiopharmaceuticals, started off with a company called AAA, and that led to an acquisition by Novartis. And of course, Novartis took that compound in the orphan indication, neuroendocrine tumors, but it works. And you've also now got a compound, Pluvicto, that works in prostate cancer. And these position very well as the last therapy. So most of these patients have exhausted all their conventional treatments, and they've been given these therapies, and they're seeing meaningful outcomes of increasing life expectancy. So that has become a very interesting one. We see that radiopharmaceuticals are no longer just being owned by the companies that used to own that space, the GEs, the Siemens, the Lantheuses, the Jubilants. It's now large pharma, who are traditional therapy space, now see this as a potential. And also, radiopharmaceuticals can be used without other drugs to do that.
Mm.
So there's been a great expansion in that. So I think, what we'll see over the next few years is major growth in PSMA, therapies for prostate cancer, and then we'll see the next generation radiopharmaceuticals emerging, and they will again be positioned late on in the clinical path of patients, but then they will slowly, during clinical trials, go to be used earlier. So it's an exciting time, especially that people are saying that the PSMA agents are, worth somewhere between 2 and 7 billion per annum as a marketplace. and back to, as you know, in China, and you can speak to this, again, it's a therapy that was well suited for China. They've got a history in radiopharmaceutical therapy. Prostate may be a sizable market, but there are other cancer indications that may be more useful in China, such as gastric cancer and things which have high presence. So you see many companies interested also in a global footprint of doing that. And I think of note, talking about other companies, Telix Pharma, an Australian-based company, they actually signed a large deal with Grand China Pharma, I believe. I might be wrong on that, but Grand China Pharma identified the potential of radiopharmaceutical therapy very early. And I've tried to bring that technology into the China footprint. And hopefully that's what we can do with our early pipeline. Similar, look to develop agents ourselves, but partner in geographies with the right partner. So again, I think for our radiopharmaceutical therapy platform, we'll be looking to partner those with Chinese pharmaceutical or companies, again, for them to develop it in the right indication for those geographies.
Yeah, actually, I remember in 2019, we visited China, and then we visited the Peking Oncology Hospital, and we talked to the radiology, the head of radiologist. They really have such eager to take some of the therapeutics to China, because back to them, I think now still they don't have such the therapeutics to treat the patient, but they see the advantage of them. They even try to develop something by themselves in the basement. I'm not so sure you remember that in the basement they have the active.
Yes, I do remember that. Yeah.
So that was a very in China, the hospital become the drug developer a lot of in the situation.
Also in radiopharmaceuticals, people state that actually it is different because you can actually image the patient.
Yes.
So actually you get a very early decision of whether the drug works, because unlike other drugs where you don't know where they go. With radiopharmaceuticals, although they've got a therapy isotope, you can image them with SPECT or PET cameras, and you very quickly know in a clinical situation, did it actually get to the target? And actually did enough get to the target to potentially kill the tumor? So it is an advantage that we have over other therapeutic modalities, that we can actually spend less, most likely to get that early decision of whether a candidate's going to succeed. So again, there are many academic groups who can do that because it's not going to cost 10, 20, 30 million to get into phase 1 and end phase 1 and get to an answer. You can do this it still requires millions of dollars, but a lot less to get to those decision points.
Yeah, that's kind of make the drug development faster at a lower cost.
Hopefully. That's what we all want.
Thank you so much for sharing your perspective of the radiotherapy treatment, and also highlights of ImaginAb. Next, please present to the audience of the ImaginAb technology pipeline and also the future plan. Thank you.
Yeah, thank you, Echo. So So I'm just going to give a very brief presentation and highlight some of the things that I discussed with Echo through these slides. So hopefully we can move this on somehow. Okay, so just, this is our standard investment and pack that we share with people. As Echo Tyler and highlighted, you can see here that we are actually a revenue generating company. We have about 26 million of revenue booked from all these pharmaceutical companies. We use a novel antibody fragment technology, which I'll step on. And we talked with Echo about the use of our CD8 imaging agent. It's actually a large market when you look at IO therapy today and people who receive IO therapy. In the US, it's about a 32 billion market, but only a third of those patients actually work on therapy. So you can look at the cost savings you can make by putting diagnostics into that. We've actually used the same technology, the mini-body technology, to develop therapeutics. It's very early, but we just identified a lead candidate to take the clinic, and I'll highlight that. And as you said, we talked about supply chain for CD8 and for other isotopes, and we've got a bit of IP and I'll show some pretty pictures of some of my staff and advisory team, and show that we do this with not many staff at all. So just to highlight ImaginAb, so yeah, we're a revenue clinical stage company. We're based in Los Angeles and in Inglewood. If you don't know where Inglewood is, and you've ever flown to Los Angeles, the plane, and we're on the landing path of most planes in LA. So you probably can wave us and look at Randy's donut sign and just turn around the corner. We focus on CD8 immunopet, so imaging of CD8 T cells, but we also have radiopharmaceutical therapy. We've been around actually 15 years, so we're getting. We're into our teenage years, but not troublesome, which is always good. But with that, we've gained a lot of experience of developing radiopharmaceuticals, which is, not unique in the field, but there aren't many companies in the world that can say they've got such dedicated staff who've got experience in the field, so we know what we're doing. Our major investors are shown here. The top three are Adage Capital, based in Boston. We've also got the Psycha Group, which is an investment firm out of Santa Monica in California, and Innoviva, which is also based in the Bay Area. We were also a bit fortunate to get investment from the Raptor Group, and actually very early by Merck Sharp Dohme, the drug company, or Merck as it's known in the US, because they saw the potential of our technology. And that's what we'll talk about in this short presentation, is about immunopet and radiopharmaceuticals. So how do you make a radiopharmaceutical? Very simply, it's a combination of three things. A binder, something that will bind to the target. In our case, it's a mini-body. A mini-body is a fragmented antibody, about half the size. And actually, it's inert. So when it binds to target, it doesn't cause any ill effect to the cell, which is quite important for diagnostics and radiotherapy. So it carries. To that, you add what is known as a chelator or a binder that can bind radioactive mechanisms. And so, adding the binder and the chelate, you get a precursor product, which is like a normal drug. You can store it and have a long shelf life. In our case, we have shelf lives, greater than 7 years, and these are used. But then you have a different step, which is you actually add the imaging or therapeutic radioisotope, and this is done in a radioactive facility or a CDMO or inside a hospital, because it's done, and once it's done, it has a short time that you can use it. For some products that are labeled with 18F, you've got to use it within hours. For others like ours, you've got to use it within a week, right? And some of those advantages. So you add either an imaging or radiotherapeutic isotope, and to that you get a final product. In our case, you get the mini-body to take it to the target, to deliver the imaging isotope so you can image the patient, or a therapeutic isotope to kill the cancer cell. And we've been doing this for many years. We've worked on many compounds, including, as we talked about, PSMA, imaging and therapeutics. We actually out-licensed our PSMA imaging agent in December last year, showing value of our technology to the marketplace. So I'm just going to skip through CD8 T cells and why people should be interested. So people listening to this webcast, if you're into IO and you understand immuno-oncology, please, go and have a coffee in the next 2 minutes. But for those who aren't familiar with immuno-oncology, really what this diagram shows, and this, circle diagram, which is a very good indication. If you look at the blue, it's got and inside the wedge, the blue indicates the number of CD8 T cells shown in a biopsy for all those cancer types. And what it shows is that if you have lots of CD8 T cells inside your tumor, then you have a good prognostic outcome. Well, why would that be? Well, actually, the checkpoint inhibitors, the anti-PD-L1s and PD-1 drugs, what they do is they allow the T cell to do its job. They unmask the cancer cell and stop the cancer cell inhibiting it. So if you have T cells inside your tumor and you can add either of those drugs, then the T cells will go and destroy the cancer cells. If you haven't got any T cells there, then maybe giving those drugs isn't going to be as effective. So you can understand why measuring of CD8 T cells for either a therapeutic company or a clinician helps them understand about treatment choice and whether a drug is working. So, following on from the discussion we had earlier, well, for anyone to use something, you've got to be novel and disruptive, but you've got to address their need. So what are the needs today, both for life science companies and healthcare companies? Very similar. They want to use methods that are less invasive. So in our case, it involves one injection, and they want to be more sensitive, accurate to either detect disease earlier or detect response earlier. As I said, also they want to replace biopsy because in metastatic patients you can't multiple biopsy patients. It is very intrusive, but it actually can lead to side effects. And they want to actually measure those changes. So why we've had success with the pharma companies is it allows them to detect whether the drug is responding and also whether the drug is having adverse events. And it allows them to look at different clinical trials designs, so maybe focused on a small cohort of patients to prove that the drug is working and actually provide them confidence to move into that. Also, many drug companies now are looking at combination therapies. So they give a checkpoint inhibitor plus a new drug, and they want to see whether the second drug adds any difference compared to how the checkpoint will work itself. In disease monitoring, patients are on IO therapy for many years, and actually it doesn't mean that it can work. They can become resistant to those therapies, very early on, after 6 weeks or after 6 months. So again, being able to know whether those T cells are still there and working is a useful tool for clinicians and also drug companies. When you look at that as a global market and you look at the number of patients that you could use this agent on, then you're talking large numbers and ranging very north of 500,000 per year in revenue. So let's just quickly step through the technology. As I said, we have a mini body technology. It has an INN name called crepmurulumab burdoxan. I'm sure you'll all remember that, and it's easy to pronounce. Hopefully we'll have a marketing name that replaces that in the future. So we have a mini body. Into that we add a radioactive isotope known as zirconium-89, and that has a decay half-life for 3 days. And we have a product shelf life of 6 days. And that's quite good for us because as we talked earlier, we can establish manufacture in all the continents. Actually, we can distribute to all of the North America from 2 manufacturing sites. In Europe, we have 2 manufacturing sites. In Australia, we have 1 manufacturing site, and that will cover all the need of those geographies. And of course, Dongcheng are also looking at a similar footprint. They don't actually need to have many manufacturing sites to cover all of mainland China. So again, zirconium-89 provides an advantage in distribution. And if someone wants to do a clinical trial with us, it's very easy for us to tell them whether we can deliver to the hospital of their choice. What you see on the image on the right-hand side and the central image, you can actually see what a CD8 image looks like. first of all, I'll go to the one on the right-hand side, which is radioactive glucose. So glucose is used by organs that use a lot of energy, the brain, the heart, but also tumors use a lot of glucose. So if you inject some with radioactive glucose, it will show you where the tumor are, and you can see a tumor in the right half of this individual. But you can also see activity in the brain. If you look at the image with CD8, you can actually see all the CD8 T cells in someone's body. So going from the top of the person, you can see the lymph nodes where you have CD8 T cells. You can also see darker areas in their long bones where you also have CD8 T cells in your bone marrow, and also the spleen where you have lots of CD8 T cells. So if you image a person without cancer, you'll get a very similar image. In this case, you can see a modest uptake into the tumor where there seems to be some CD8 T cells. So we'll go on to that, and what does that mean? So first of all, I'll just go over one of the value propositions. So this is just a pictorial showing that if you wanted to biopsy someone who's got metastatic cancer, you'd have to put that needle into many different tumors to understand whether each tumor is being affected by the treatment. Also, you have a sampling error because you're actually inserting a small needle. So you're only sampling less than 1% of the tumor. So you don't know if those results are really representative of the whole tumor or what's happening in the body. If you could image someone, as we show on the picture before, you actually get a global picture of every lesion inside their body. And you can actually look at that globally, or you can interrogate each lesion. So we bypass some of the intrinsic issues that you see with biopsy about detection of CD8. So this is an example, and is what excites drug companies and biotech companies. And let me just spend a little time on this. So again, we've got an FDG image, the first image you can see on the whole body image, which we showed before. And you've got the two images in the center where we've imaged CD8 T cells. So we talked about the first image. So this patient is imaged before they had treatment. So on baseline, before they received any treatment. And you can see, and I'll try and use my cursor, you can actually see modest uptake into the CD8 T cells. This person received one cycle of pembrolizumab, so Merck's drug, also known as Keytruda. And actually you can see you can see a uptake that's darker inside the tumor. If you actually go down to the bottom panel, what's interesting is this is a cross section through the leg, and I hope you can see it, that you can see a bright red ring around the tumor. So in the field of immuno-oncology, that's an immuno-excluded tumor. So the T cells are outside the tumor trying to get in, but they're not going in there because they don't know what to kill. Once they're given Keytruda, which stops that inhibitory pathway, you can actually see a more intense and homogeneous image. So in this case, you can show that the checkpoint inhibitor is starting to work. It's actually allowed the T cells to go in and start doing their jobs. What's really fascinating is when you look at the X-ray CT images on the far side. These images with our agent were done at 1 month. If you look at the 1 month image of the CT and compare it to the baseline, the tumor is the same size. If you'd used RECIST at 1 month, you wouldn't have known Keytruda had worked because the tumor is the same.
Yeah.
It took them over 4 months, 6 months to see that there was a reduction in tumor size. So what we're doing is we're actually telling people this early engagement of their drug a lot sooner than they can use using conventional methods. And for some people, that's very powerful. It allows them to make decisions. In this case, they could have made a decision 3 months early.
Yeah.
And if you look at that as a cost-effective clinical trial objective, you can see why people use our technology. We talked about this in the highlight, but it is important. We'll go from the bottom first because Echo's in the room and MSQ, and we did the deal in May 2022 with Dongcheng, and that was great. One, because it actually allowed us to, an external party validated our technology. They went through extensive due diligence, and also we had a very good deal structure then. We agreed the model in China. We agreed what the value of this is, and we agreed a deal based on future revenue and milestone payments. So, you know, it allowed us to get confidence and show the rest of the world that someone believes in our technology. And actually, Dongcheng have got great experience in radiopharmaceutical therapy. So it's a great partner for us, one, because they understand technology, and they've got the ability to execute and deliver on that technology. So if you look back to our journey in 2018, when we signed our first deal, we'd only just got our first images. So those companies such as Boehringer Ingelheim and Merck, you know, took a leap of faith with us on very early data because they knew they had problems and wanted to see those images. But as we evolved our clinical trials, when we got more safety data and completed our phase 1, we could show that data to other pharma companies, and that led to more deals in 2019. And that was followed by our phase 2A. And we did that in collaboration with 3 pharma companies, Takeda, AstraZeneca, and Pfizer. They got early access to the data, allowed them to feel confident to sign multi-project deals with us. And we continue now in our phase 2B, and our goal is to seek approval late 2024, 2025 with this technology. And this highlights all the partnerships that we have. It's slightly out of date, but as Kaseko keeps telling us, we have a press release every so often, and I think there's one missing because we've just done a deal in the last month, and we'll probably present that at the ACR and ASCO. So as you can see, as in here, we evolved, starting off with Boehringer Ingelheim. We really started with large companies, and then it came along with all the biotech companies becoming interested. And as you can see, we actually use this in multiple therapy classes, T-cell engagers, all the way through to non-oncology indications. So we actually have imaged people in COVID-19, and we hope that will be published pretty soon. But we're also working with a company called Apcuro, who are actually looking at in muscle wastage disease, where T cells can cause muscles to get atrophic, and their drug actually stops that process by halting CD8 T cells. So we've started conversations beyond oncology with certain companies, and there may be a growth there, especially in vaccine development, because of course vaccines cause the CD8 move. Our deal structure, you know, is very simple. We have a access or license fee. We provide project support because some companies are not familiar with imaging, some are. So we do that at cost. You know, we just make sure they do that, and then we charge them a manufacturer. So every time we make it for them, because we make it bespoke, we put their clinical trial and sometimes their patient ID on that. We make it to order, then we charge them a dose for every time we make it, and a delivery cost because it depends where they want it shipped. So that's what we do, and we support those companies from start to finish during their clinical trials. And we have a team also in Europe because we do clinical trials in Europe, so they're on the right time zone, so they can answer any questions immediately for those drug companies. So as I said, we expect to get approval in 2025. It's a large market. It's actually high margin. You know, radiopharmaceuticals have a high margin. You know, we shouldn't be scared to talk about that. You know, the key for us is to have a low manufacturing cost and distribution cost, allowing us to make a return on the investment for our investors on all the money we've done. As I said, you know, we've signed more than 19 contracts now. That's greater than 20 contracts, and we've booked in over 70 doses for the next 2 years to be delivered. And how we've done that is by, you know, also negotiating in those deals upfront payments, which have reduced our need for diluted funding. So we balanced how we strike those deals to get cash in through the door now that stops us needing to raise money. So also that's how another trick that we've used to ensure that we don't need to seek cash all the time. I'm conscious of time, so I'm just going to switch technology, and this is what was highlighted as a key emerging area, and it's worth reading about. So RPT, radiopharmaceutical therapy. You know, you can type that into the internet and you'll get some great articles. So if you ever wanted to develop radiopharmaceutical companies, we asked the market, what do you think you need to be successful? And we came up with these 4 parameters.
You've got to have access to some proprietary molecules or technology. You can't just use generics. You've got to be able to identify both the targets to go for, but also the clinical addressable market. Where is the highest unmet need? You've got to have a team that can actually implement this work. And what's important is that you have addressed the supply chain. So have you got access to the radioisotopes to be able to do this? If you haven't, you can't deliver it. So you've got to be able to think very early on about your supply chain, and that there are no freedom to operate issues, or even in the modern world, geopolitical concerns, you know, where you do deals can sometimes change because of change in political power. So why do we use mini bodies? Very simply, there's an advantage. So we call it the Goldilocks technology. It's not too hot, it's not too cold, it's just right. So really what we do is we address some of the fundamental problems with large antibodies. Large antibodies are very specific, but they hang around too long in the body. So you don't want to have radioactivity circulating the body and giving unwanted dose. So what this slide really shows is that we can maintain high tumor uptake, but clear from the blood very quickly. So we have a perfect balance. And so this is, as I said, it's the Goldilocks technology. So with smaller molecules, sometimes they have poorer tumor uptake and clear too fast, and antibodies hang around too long. So what we do is we tune mini bodies to have the right characteristics dependent on the cancer indication we go for. And this is a very complicated slide, but it tries and addresses a few things. So this is the PSMA story, so radioactive PSMA. And you can see it looks at three technologies: antibodies directed to PSMA, and small molecules directed to PSMA mini body. And very simply, you can see on small molecules, you can see from these images, look at the green box, that you can see lots of little dots in the person, and that is the agent binding to the metastatic tumors in that patient that are PSMA positive. And that's really good. But if you could also look, the blue circle also shows non-specific uptake into the salivary glands. So in these patients, you can actually ablate the salivary glands, which actually is not a very good side effect. The other issue with small molecules is they clear via the kidneys. So you're pushing a lot of radioactivity through the kidneys, which can cause or mean that you can give less activity because you have to protect the kidneys. So what we're looking to is to try and address that. So antibodies and biologics are more specific. So you can see from the antibody and mini body pictures that you don't have any uptake into the salivary glands, and actually they clear differently. They clear via the liver and the kidneys. So if you can look at those images, you can't see the kidneys. So you're taking some of the radiation burden away from the kidneys. But also, you know, we keep the specificity. So by going to a biological, we do that. And our thesis is with a mini body, we can deliver it the same as an antibody, but clear it from the background, which means that we may have a potential to be more safe. And also that you want to actually match the isotope that you're using with the molecule. So as an example, lutetium has a half-life that's very long, you know, greater than 7 days. So if your molecule hangs around, it can hang around in the tumor greater than 10 days. If you put lutetium on it, it'll have more killing power. If your compound goes to target and clears, actually you've excreted all the radioactive killing power very quickly and not leaving enough in the tumor. So there's that balance there. So this slide really, it focuses on what we work on. So our goal is just to look at clinical indication, and our clinical indication, we pick them based on a number of things. One, there's limited competition from all drugs, and actually the survival rate of those patients is still poor. And that's highlighted by the ones highlighted in green. So as an example, small cell lung cancer, not many drugs being developed in that, a very difficult proposition, and the survival rate at 5 years is 6%. So we feel that's a great opportunity to place radiopharmaceutical drugs. And so that, as you could guess, is an area that we think is of interest. And that is the same for ovarian cancer. Again, you know, these patients have poor survival in the long term. So, when you look at it, our key programs that we're looking at are small cell lung cancer and ovarian. We're progressing those. We've took those candidates. Actually, we've selected a lead candidate for ovarian cancer. So this slide, is actually more advanced for now the ovarian cancer, and our goal now is to find a lead candidate for the small cell lung cancer. And our goal would be ideally to complete pre-IND studies in 2023, early 24, and go into the clinic in 2024. As I said earlier, we historically worked in PSMA. We actually out-licensed the imaging agent actually in December last year, but we are looking for licensing partners globally, either in specific regions or in a global license for our RPT prostate cancer agent. So, if you're interested, you can contact me or Echo, and we'll be happy to talk to you. This slide just tells you where we're on our path. I think the key thing that shows you is we're very rapid in what we do. We selected our candidate in Q4 2022, and it took us just over a year from idea to selection to get that one. So with the team, we tried to go through a rapid development of engineering of mini bodies and cyst bodies to lead confirmation and start the work in therapy and models to prove that those before selecting to go to the clinical indication. So here's a highlight of some of our leadership team. I'd like to start at the bottom with Anna Wu, who actually founded the company ImaginAb 15 years ago. And if you look at experience, she is actually the professor and chair of the Department of Immunology and Theranostics. She's got great experience in everything that we do. So she's a great board member for us and scientific advisor. And it's always a pleasure to work with her. And then we have Christine, who has a lot of experience in radioactivity and nuclear medicine, and has worked in many industries. So, but as a clinician, she can help us identify the areas of unmet medical need. And we have Gareth Smith, our VP of Business Development, who worked at GE, and that's where we met. But he's also most recently worked at a radiotherapy company called Theranostics. And he has been fundamental in establishing our radiopharmaceutical pipeline, and that we've supported him to move this as a new aspect into the company, along with our Director of Research, Alexandra Marcioni. And last but not least, we have our Chief Financial Officer, David, who makes sure that we keep an eye on our finances and keeps us all under check. I just wanted to highlight with this slide, actually we're a small team. We're only about 27 globally, so we get a lot done. We hire staff on the things that we think we need to do, and we outsource the things that we think other people can do better than us. And when we outsource, we make sure we've got people with experience to manage those. So we're 27, majority of the staff are in LA, but we have a clinical group based in out of Dallas, good hub to get on a plane, to travel the world. We have a small team just outside London in the UK, and then we have a sales team within Europe and then on the East Coast and the West Coast. So we are small but covered in those geographies. And as you can see from our advisors, that we use these advisors to help us understand our radiopharmaceuticals, and we're going down the right path. So in summary, I'll return to the first slide, just to show that we're a revenue generating company with a focus to deliver the CD8 imaging agent to the market by 2025. We continue to push that with pharma and biotech companies to generate revenue, but we are developing a radiopharmaceutical pipeline as our next candidates to take through and grow the company. So thank you. I think now we're going to have a closing chat. Is that right, Echo?
Yeah, so we do receive the several questions. I know you have to go to airport soon to catch up the flight. Thank you for this great presentation, and also very excited the progress you made and also for the future plan. So I will just quickly go through probably 3 to 4 questions. Okay, I will let you go at 11. It's important to catch the flight. And so you are talking about the pipeline candidates. So when you select the indications, you have showed the slides like less competitive, right? So, however, what do you see? The peptide candidates can be unique, and how do they different from existing the treatments for the same indications?
Yeah, so as we said in our early discussion, we're positioning RPT in patients who have exhausted their current treatment. So you're positioning post any other treatment. So as an example, if you look at ovarian cancer, most patients could enter a maintenance therapy regime, and hopefully that works for a long period, but for some, it stops working. At that point, they have no other option.
Okay.
So our positioning is to put a radiopharmaceutical therapy at that point when they've exhausted all their other options. And from that gets an initial indication. And we hope either on its own or in combination with other drugs, we can move it earlier into the care path of that patient. That's the same with small cell lung cancer. It's an indication that it's hard to treat. There aren't many drugs that work there. And those patients, unfortunately, their cancer can accelerate very quickly. So for them, we see those as ideal indications for us.
So that's really the save life.
Yes, it is. And the first is to extend life. Yeah.
So can you discuss a little bit more about that preclinical and clinical data that you have for these pipeline candidates? As you mentioned about, actually you collect a lot of the safety data through the partners.
Yeah, for the CD8 imaging agent, we have, we've got, hundreds of clinical data points, that have been done by us or by pharma companies, and that's important for us to assimilate our filing. So, we've got extensive data. Our agents are shown to be safe. We haven't shown any adverse events to our agent, touch wood, to date. And we've shown that we can manufacture that and produce the same product in three continents, which is also important, showing that we've got robust information. On the radiopharmaceutical, as I said a lot earlier, it's really around two points. Can we show that the agent is taken up into a tumor? And if it's taken up in a tumor, can it kill that tumor? So that's where we are with both the ovarian and small cell lung cancer. We've got data showing that our candidates have the ability to kill those tumors and extend the life of mice. But what's also important is to show that they are safe. So in my presentation, I showed about, normal organs can become toxic. So we studied the distribution of our compound, and our data shows that the uptake into radiosensitive organs, such as kidneys and other organs, is not an issue for us. So, giving us confidence that we should be able to administer a safe dose in a clinical situation.
So what are the biggest challenges you are facing developing this treatment? It's because it's very novel, and also it's not really you have so much the example you can follow, right?
Yeah, so as we said, we know the team, and we know what a good radiopharmaceutical therapy agent should look like.
Yeah.
So we have success criteria that we can develop, and we know from other agents that have been developed, Lutathera, Pluvicto, and others in the past. We know what makes a good agent and what makes a bad agent. So we have set internal criteria to be able to do that. I don't know if that answers your question, but we feel confident that we can select those, and actually that a mini body can provide a unique solution compared to maybe small molecules and large antibodies. So we feel we've also got a place to provide a different platform technology for candidates.
So follow up this. So I think this is how do you envision your pipeline evolving over time? Like just you mentioned about actually you even expand beyond oncology and the vaccine. So what are the new areas of research you are exploring for the future development?
Yeah, so for CD8, we're very blinkered internally to try and get approval in the oncology indication.
Mhm.
But via partnerships, we've shown that it has got other potential. We've done a study in COVID, and that is become interesting to some people working in the vaccine area. But we've also started to look at other indications where CD8 could be a potential, such as there's been research recently that's shown that CD8 T cells may be implicated in multiple sclerosis. So, you could envisage a partnership with us, with someone who wants to explore that as a potential. So, with CD8, we will drive it to the market and then expand indications post-market in the right indications and look at that with a commercial eye. In radiopharmaceuticals, I think we'll stick to what we said. We'll focus on areas of great unmet medical need where drugs are failing, and unfortunately there are plenty of cancers where that is. And our focus is also on validated cancer targets. We're not a discovery company looking for something new. We want to be able to explain to someone, here's many years of research regarding this receptor. A bit like PSMA, people accept PSMA as a valid target prostate. We want to do the same. We want to generate radiopharmaceuticals to validated cancer targets that people already understand the biology about.
That's great. Because of the limited time, so I would say thank you so much, Ian, for taking time to join us today and sharing your great insights on the ImaginAb's technology pipeline partnership, and also your secret sauce for developing the company to be the cash positive. I think that's really the key for today.
Thank you, Eka, and also thank you for all your support over the last few years and opening our eyes to other potentials. Thank you.
Yeah, thank you. I think actually I wanted to acknowledge your entire team have been very lean team and work so hard and make so much progress, and also the pressure of us to work together and develop the success across the board.
Thank you.
Thank you. So I want to also thank you for the audience for tuning in and for the thoughtful questions that were submitted. So we hope you find today's discussion informative and valuable. If you have any further questions, please feel free to reach out to Ian or us, and so we will get the conversation continued. So thank you again, Ian, and our audience for joining us today, and have a great day and have a great trip back to Los Angeles.
Thank you very much, Eka. Bye-bye, everyone.
It's such a pleasure to be with you in person. Thank you. Bye.